These three things is the autistic phenotype. You name me, you go find me one autistic phenotype with that does not have problems with the central nervous system, the peripheral nervous system, and the enteric nervous system. You can't. You can't find it. Here we go. Okay, this one's going to be dense. This one's going to be fun. This is Neurulation. We're going to cover specifically 2 episodes we did on, we'll just write them down here. 56. It was called Autism, Neurulation, and Autism, Neurulation, and Embryogenesis. Episode 57, Neurulation, Neuroepithelial cells, and Mesencephalon, which is a type of neuroepithelial cell. Ooh, I'm close. We'll make it. We're going to make this. Tryptophan. These 2 kind of will branch off out of these 2. Nicely. You'll see, hopefully. And then way back in an old episode, we really went into kind of embryogenesis to kind of perinatal, just after birth. I think maybe up to 2 years. I can't remember actually, but we spent a lot of time. UC San Diego, Ivory Tower. They spend a lot of time in this little epoch. It's great stuff. They have great data. That episode really went into kind of artificial light. We're going to get there though. So we're kind of going to be talking about these 2 episodes mostly. So I just spent a lot of time writing those episodes out. I probably shouldn't have done that. Just stick to just kind of like the objectives of the information. So we're going to talk about neurulation and embryogenesis. And this is the neural plate, okay? So this comes on board around day 21, 23 or so. This is what we are to begin with. The glare is okay, I think. Neural plate. So we got the epidermis in the blue, ectoderm. Neuroectoderm is here in the middle, and then the little green's called a border. And then this thing folds. See, this will fold in, and the neuroectoderm will come down. And this is, we have different types of derm now. Ectoderm is at the very edges. The mesoderm is the next layer kind of in, and the endoderm is the center of this fold. And this is where skin and nervous systems will begin. Skin and nervous systems are like that. And then we have the tube, because we're folding into a tube. And we have the epidermis. It's growing larger, okay? It started here. We have the epidermis on top. And then the green are neural crest cells. And then we have the tube, which is right here, the neuroectoderm. So when Jack Kruse says things like the skin and the oral tissues and so forth, and the central nervous system are combined. They're like that. So this tube will go on to develop the brain and spine, the central nervous system. These neural crest cells, they're just kind of drawn as circles. This is where already on board melanocytes are being created. Remember, because of the skin. And then the adrenals. Our adrenals, we think of things like adrenaline and cortisol and so forth. And then face and bone and cartilage. And then here, the peripheral and enteric nervous systems are beginning. So this is more central nervous system, and this is more peripheral and enteric nervous system, okay? Now, there's some key components here that are very worth mentioning, and we've covered these. So just kind of recapping these. SHH. What is this? We have this. And it's huge in development. Sonic Hedgehog. This is involved in proliferation. So we're going to be making cells here, right? We make a lot of cells here. We proliferate cells. Cells divide. It's kind of just the process. Progenitor cells is big with the Sonic Hedgehog. Progenitor cells and cell growth. and Sonic Hedgehog will also control cell survival. Sonic Hedgehog is involved with motor neurons and interneurons. What are those? We could simplify neurons in the central nervous system as sensory neurons, interneurons, and motor neurons. If we want to really have like a simplistic view of things, sensory neurons, they're involved with sensory input. Interneurons are things that are connecting regions, things like excitation, inhibition. And then the motor neurons are responsible for things I'm doing right now, just moving. I'm constantly moving. EI is a big one here, from here. Okay, now if you look at data, research data, and you can find like mice and rat models of autism. If you knock out, it will look like this. Sonic Hedgehog knockout. The mouse or rat will not develop the thalamic reticular nucleus. This part comes from the ventral thalamus. Okay, so if you knock out this in a mouse or rat model, they will not develop this or that. So this must be a region of interest for us because of this. Remember the part of albium, interneuron specifically. And then this. We've covered that in an episode about inhibition because that's a sensory gate. It protects the thalamus. You don't want the thalamus to be overran. Insane. Okay. We're getting there. Shoot, we're already 9 minutes in. God bless it. Next one is P10. Phosphorase. No, that's not right. Phosphotensin. For sure it's tensin, but I don't know how to spell it. Let me check my note. That's it? Insane. ACE. Anytime you see ACE, it's an enzyme. So this is an enzyme. P10. It's huge. This is going to be huge. This is a lipid and protein phosphorus. I can't spell that. I can't. Lipid and protein phosphorus. All of our membranes, except for the inner mitochondrial membrane, are lipid phosphorus. Every cell membrane. This is huge. This is also involved with regulating neurulation here, in large part. Okay, there's a pathway. Let me see. Yeah. There's a lot of PIP. You'll see a lot of PIP involved, like 2, 3, and so forth with this. But let's not get distracted here. AKT and mTOR, mammalian target of rapamycin. Okay. This is huge in this process. Okay. Especially the neural plate. Okay. This is going to be such a region of interest. This is also involved with, and because it's lipid and phosphorus, microtubules. We have an episode on that. I wrote it out. And mitochondrial biogenesis. Okay. There's a study recently, Eric et al. In 2021 or 2022. I can't remember. But it's in Cell Press. Cell Press is a fantastic journal. It's actually maybe Cell Reports. I can see the graphical abstraction. Cell Press journals have this great graphical abstraction and then like 3 or 4 key highlights. It's very good stuff. Okay, so they study that a loss of P10 and then mTOR complex 1. You'll see it as mTORC1. I forget what they did. I forget if they rescued the lack of P10 by targeting complex 1 or what. I don't know. But this is a good region of interest here for us. This regulates cell growth, proliferation, survival, and differentiation as well. And the proliferation here into differentiation into a mature cell. P10 is also involved in things like cancer growth and things like that. Okay, now we're getting somewhere, I think, because we got to get to the mesencephalon. we got to get to the mesencephalon big time. And also, and we won't spend much time on this one. B9. Okay. Methylene tetrahydrofolate reductase. No, what did I say? Methylene tetrahydrofolate reductase. Methylene. This is the motherfucker gene. And this is B9. This is leucovorin. Okay, I'm not going to sit here and explain this because you can go see Richard Fry and Nicole Rincon. Probably the best in the world. Best in the world. Former guest on the podcast. Best in the world on this. This folinic acid and leucovorin. Okay, this is building block though for protein synthesis and so forth, and DNA methylation. This is a big thing. The DNA methylation. Remember Richard Fry? We talked about how leucovorin is helping possibly the distal connections, like from the superior colliculus to the frontal eye fields, is a very distal connection, or the superior temporal gyrus, sometimes Wernicke's and so forth, to Broca for the speech and language. This is going to be a big thing here. It's also involved with methionine. It provides SAM, the donor or cofactor, to methionine. Methionine and tryptophan, which we're going to talk about now. They are fantastic amino acids because they only have one codon. Tryptophan and methionine are so special. You know this is an aromatic amino acid. This means it needs UV light. Aromatics are UV light antennas, essentially. They have benzene rings and pi electrons and things and so forth, which helps it synthesize. Tryptophan is like around 277, 283 type of peak absorption, which is right on the line of UVB. UVB starts at 280. But this is huge because maternal peripheral tryptophan equals brain tryptophan serotonin. The mother peripheral tryptophan is how the fetal forebrain gets tryptophan and serotonin. Nobody does anything with this stuff though. I'm telling you, nobody cares. Nobody does anything with this stuff. This is involved with those sensory maps. Serotonin has massive roles outside of mood, outside of, it's so unbelievable to me. Serotonin has hidden roles, okay, with receptors and cell differentiation and proliferation, with initially overgrowth of neurites. Neurites are axons and dendrites. How much information do we need about autistic phenotype having abnormal axon and dendrite? I don't know. Especially in the central nervous system and the peripheral nervous system. Serotonin. It's unbelievable to me. The mother's tryptophan levels. Remember, tryptophan being that aromatic amino acid, if it's under hypoxia, okay, so you have tryptophan, and that synthesizes serotonin, and then some melatonin. That's melatonin. Okay, oxygen levels will move this way, depending on your oxygen levels and ROS, things like that. So it depends on the oxygen and so forth will depend on how the tryptophan's being synthesized or not being synthesized. Nobody cares. I don't think. I don't know. I don't know why things like this is this so foreign in autism research. Okay. Anyways, so once we get here, we're going to spend most time here for the central nervous system. So there's four cell types that will come on board: neuroepithelial cells, which will become 4 cells as well. And then, let's see, the neural crest cells will begin to do different things. The ectoderm, okay, so kind of it's just expanding out here, and then mesoderm, okay. It's just, we're this, and then maybe like that, and then we fold, and then this another layer comes on, and then we're just eventually becoming, we're just expanding. It's almost like a finger looks better because we're about to draw this out right now. We're about to draw it out. We're just going to get rid of this because we got to get to the mesencephalon. We got to get to the mesencephalon and these epithelial cells. So we're just going to go here, okay? Because we're developing the brain here, brain and spine, central nervous system, okay? So I need different colors here, I think. I think that's what we'll do. Now, red's going to be mesencephalon, I think. We'll have to do red for mesencephalon. Okay, so we have the proencephalon. Remember, there's 4 different cell types. Proencephalon, mesencephalon, and then we'll go green. We'll go green for the rhomencephalon because it's a lot of going, a lot of reflexes. And then the 4th one, to be honest, it's making the spine. It's making the spine. So right after that fold and tube and the epidermis coming on board, we kind of start looking like this. Okay, this is what we're kind of just looking like here. Okay, this is pro here, mesencephalon here, rhomencephalon here, spine. All right, so these will begin to subdivide. So the proencephalon, the forebrain, goes into, I hope you guys can see this, telencephalon and diencephalon. Okay, so essentially this one is the cortex, this one's the subcortical areas. Okay, and then we'll start to look like this. It's almost like a heart. Maybe I'll draw it keeping with this theme. Okay, so we'll start going like this. Okay, it's getting bigger is what we're doing. This one, if you follow along with the podcast and me ranting about this all the time, nothing. This one will make the mesencephalon and the myencephalon. So this will become the cerebellum and pons. And this one is the medulla. Okay, so let's see. We'll start looking like this. Okay, that's good. That's good there. So this is the proencephalon. Okay, it's just the big head. It's the majority of our brain is right here. This is the smallest part, and then this is the bottom of the brainstem, or most of the brainstem, and then the spine. Okay, so let's draw this out as the brain. All right. So the proencephalon. So it might be easier to go bottom up. I'm just going to go bottom up. So we have the brainstem here, the start of the brainstem, the medulla. Okay, this is the medulla. And then, let's see. So this is the myencephalon, okay? And then the mesencephalon, the pons, is a little football oval shape that sticks out in the front side of the brainstem. And then we also have deep inside here, it's kind of covered. Sometimes you'll see it covered with things like the occipital, temporal, and sometimes you won't. It just depends. But I'm just going to cover it. And then the mesencephalon, okay? I got to go up higher because the mesencephalon is not very big. The mesencephalon will be like that. And then I need blue. I need blue here. Okay, I got to finish this out. We might just cover it though. So we have the occipital lobe, then the parietal lobe, that sensory motor area. The frontal eye fields will be here, and then we'll get into the prefrontal cortex. Then this kind of it just kind of comes in, and then the temporal lobe will kind of do this thing right here. Okay. Then like your eyes will be here, retina, your eye, things like that. So this is responsible for all of this. This is only responsible for here. And then this is key areas, a lot of autonomic functioning right here. All right, so let's talk about the big thing here. Oh, this is way too long. I gotta clean this stuff up. Let's just go ahead and draw this out because this is the whole part. Everything about autism might be able to be explained right here. Everything during this timeline too. Okay, we talked about how the central nervous system, the peripheral nervous system, things like the oral microbiome. The first time Richard Fry was on the episode, he mentioned like a size of a one of the teeth. Oh goodness, my teeth thing. I'm not good with molars. One of the molars, the size of a molar, will predict autism. Richard Fry told us that. And AJ Drexel was on that study. Kristen Lyle was on that study. And that's what connected me to Kristen Lyle, who was on in December. Kristen Lyle, Modifiable Risk Factors with Autism, received one of the new NIH grants for investigating autism. Okay, let's get into this. I'm going to draw what you might think is a butterfly. Okay? I'm going to try to do this good too, because this is going to be hard. No, yeah, that's not so bad. We're looking straight down into the mesencephalon here. Okay, so we removed the whole top part of the head and we're just looking straight down into it. All right, so we're going to have different regions here, okay? I should have used different colors. It's fine. This dark spot here is the substantia nigra. Substantia nigra, which translates into black substance or a substantially black substance. Okay, this is the mesencephalon can be divided into three main parts. Okay, this is like a not pre. It's like a I don't know, cerebral something. I have no idea what this area is, to be honest. And it's right here, the front part. Okay. And then we have, might as well make it red nucleus. It's involved with motor movements, different types of motor movements. Okay. We'll go blue for a lot more of this stuff. Here, medial geniculate nucleus. Okay. This is actually part of the thalamus. This is where auditory goes as a relay station for the thalamus. Because this brainstem coming up, the brainstem is going to be heavily involved with auditory, sensory, and we're going to get to the sensory part. Okay, I'm just kind of just drawing this stuff out now. I want brown for para-aqueductal gray. Okay, let's see. This part is going to be huge because we talked about this part quite in detail. Superior colliculus, eye movements. Eye movements will go right here. Once it hits the lateral geniculate, these will send some fibers down to here, and this will go back up and recruit the frontal eye fields here, and it'll just be like a distal connection, and it helps with modulating our eye gaze and eye contact and things like that. So, this is also receiving inputs just like visual cortex 1. So I think it's probably like identical timing after the lateral geniculate. Okay, so then there's also some cranial nerves. Okay, there's some cranial nerves. This is ocular motor, more eye movements. I'll go over here. A thing called Edinger-Westphal. This is kind of some eye movements too, but there's some choline involved here. This has like two parts to it. Okay, one is the choline, and it will send information down to ganglion fibers, but this has indirect influence with what am I trying to say? Pupil constriction. Because I was thinking about the light reflex, pupillary light reflex, which is a hidden danger. It's a very hidden danger with LEDs and blue light. I think this is going to be hitting hard. Because if you think about it, too, it's sending ganglionic fibers down the spine, which is full of ganglionic fibers. And our eyes, with the constriction and dilation and so forth, with these light flicker that we cannot see, but it's happening. Okay. And then ocular motor. So there's massive amounts of um, eyes going on here. And there's some cranial nerves. Cranial nerves 3 and 4 are here. That hole right there, I think this is where cerebral spinal fluid comes up from the spine and then into the brain. Because right on top of the mesencephalon is this thing. Ugh, that's awful. That it looks like a talon, and this area right here is full of water. There's not a lot of blood-brain barrier here. Looks like a talon, and all of these surfaces are surrounding this. Okay. When you see things like this in the middle of the brain, like the caudate nucleus has a long shell. It's like right here. You can see these things. There's like a fornix that's extending off from the amygdala. It just depends on how lateral you are, but you can see it like this. It's a paraventricular, ventricular area right here. Lots of water. Lots of water. So this is huge because the mesencephalon here, I'm just going to get rid of this stuff because the mesencephalon has 3 main roles. Motor movement, which is the red nucleus, but mainly here, because this is broken down into 2 parts. Pars compacta, which is both dopamine. This is huge. This is the basal ganglia, okay, and Parkinson's, because I'm going to draw something out about Parkinson's real fast here in a second. And then the other part of the substantia nigra is pars reticulata. That reminds me too. The reticular formation is very much involved here. The reticular formation is, if we just draw the brainstem again at the start of the cerebellum, and then the mesencephalon is here. What was I talking about? I forget what I was talking about. But the pars reticulata is GABA. Okay, and this is synonymous with globus pallidus internal. The reticulata and globus pallidus internal are kind of like output areas of the basal ganglia, which is regulating movement, direct and indirect pathway. We'll do whiteboard episodes on that because it's heavily involved in Parkinson's, man. So bad. What was I doing here? I don't remember. But if you can, never mind. Sensory integration. Okay, multiple forms of sensory. You have the cranial nerves here, right? You have the superior colliculus, which is receiving eye movements. You hear a sound over here, and you're just it's just reflexive. This is a very powerful area. And like barn owls, it's called optic tectum. Great research on that with Eric Knudsen from Stanford. Go see his stuff, man. He has great papers. But this is also incorporates sound, right? Because if you hear a sound to your left and you automatically look, you can pick up that target pretty accurately. That's so reflexive. And then overall, this little area here is orienting attention. But what kind of attention? To the outside world. All of this stuff is receiving information fast from the outside world and then kind of recruiting distal areas, right? Substantia nigra with the basal ganglia, eye movements, hearing. PAG is big with pain perception and just kind of, you've probably heard, I know you've heard of fight, flight, or freeze. When we freeze, the PAG receives information from areas like the hypothalamus and probably the habenula and the amygdala, the paraaqueductal gray. when we freeze, remember there's a lot of ganglionic fibers running down the spine from this area. This thing is shut off. This thing shuts the spine off, and it just shuts your motor movements off. It's so powerful. This area right here is so powerful. But this right here, these three things is the autistic phenotype. You name me, you go find me one autistic phenotype with that does not have problems with the central nervous system, the peripheral nervous system, and the enteric nervous system. You can't. You can't find it. This is also involved because I mentioned the reticular formation with sleep, the mesencephalon, and even this brainstem. So here we go. The reticular formation is just a collection of areas here within the brainstem, just like different areas, and it's has ascending and descending fibers. The reticular formation is big with this. Of course it is. So let's talk about that picture of the Parkinson's, because you can look this up. This isn't hard to find. Okay. So I'll do brown because it's supposed to be like tissue. Okay. So let's draw this out real fast, just real loosely. It doesn't matter how good it is or accurate. Okay. So we have this cerebral uncle thing. Then I want to draw another one. Okay. And then the substantia nigra here, right here. This is Parkinson's. It's black, remember, because it has neuromelanin in it. It's loaded with neuromelanin. These areas right here, because it's full of water, melanin in water is our energy source. This is biological energy. The blood-brain barrier here is very weak because there's so much water. There's so much electrical power coming through here. So with healthy people, the substantia nigra is dark because of that neuromelanin. With Parkinson's, you can barely see tissue. You can barely see it with Parkinson's because their neuromelanin is gone. And this, and this, and this, right here, it's controlling movements. You think about the autistic phenotype and stereotyping. Stimming. We'll do some more stuff on the basal ganglia. This little epoch of time is so underrated. I feel like so much of what we talk about is just so underrated. Is that because autism research just finds things and nothing is followed up? But mesencephalon. This is why I'm so fascinated with the mesencephalon.